Hello everyone, this is a continuation of the previous video on extracellular vesicles, and today we'll be looking specifically at red blood cell derived vesicles and their applications. Now, these red blood cell derived extracellular vesicles are also a type of EVs, and they're essentially small spherical fragments released from red blood cells. They're lipid bilayer enclosed vesicles, and they're a part of a larger class of extracellular vesicles. Now, these EVs are particularly interesting due to their origin from the red blood cells and their lack of nuclei and organelles in their mature form. These RBC EVs, extracellular red blood cell vesicles, are primarily classified as microvesicles, which typically range from 100 to 1,000 nanometers in diameter. They're formed through an outward budding and fission of the plasma membrane of the red blood cells, as we have discussed in the previous video. Now, this process can be triggered by cellular stress, aging, or any other changes in the intracellular environment of the RBCs, such as, for instance, calcium influx. The composition of these EVs reflect their cellular origin, which contain various biomolecules found in the parent RBC, including, for instance, lipids, for instance, phospholipids and cholesterol, which make up most of the vesicle's bilayer, and proteins, integral membrane proteins and cytosolic proteins. Hemoglobins, for instance, the proteins that are responsible for oxygen transport in the RBCs, can also be detected in these vesicles. And besides that, RNA as well. While mature RBCs do not necessarily contain DNA, they contain various forms of RNA which are included within the vesicles. Now, these RBC EVs hold great importance in a range of areas, such as, for instance, coagulation. These RBC EVs contribute to the blood clotting process by providing a phospholipid surface that can support the assembly of clotting factors. These factors are crucial for the coagulation cascade, which is a sequence of processes that ultimately result in the formation of a blood clot. Now, the phospholipids on the surface of the RBC EVs offer a platform where important clotting proteins can collect and also interact, which accelerates the formation of the clots. Now, this function is vital for not only stopping bleeding and initiating the wound healing process, but also it can play a role in the pathological conditions where clot formation becomes excessive, such as, for instance, thrombosis. Another application is in intracellular communication or communication between cells. Now, these vesicles aren't just passive carriers of biomolecules, but they rather actively facilitate communication between cells. By transferring their contents, which can include proteins, lipids, and various forms of RNAs to different cells, RBC EVs can influence a range of cellular behaviors, such as, for instance, angiogenesis, which is essentially characterized as the growth of new blood vessels from pre-existing ones, which is crucial for healing and development, but also plays a role in tumor growth, and apoptosis, which is essentially characterized as programmed cell death, a process that's essential for the removal of dangerous or damaged cells. Besides that, it's also used for cell proliferation, which is a process by which cells grow and divide, and this is critical for growth and regeneration. Besides that, these RBC EVs have great potential as diagnostic and prognostic biomarkers. The levels and the content of these RBC EVs can vary significantly in the presence of various diseases, which can make them valuable biomarkers. For example, in cancer, RBC EVs may show altered levels and compositions that reflect the tumor's presence or progression. For instance, the presence of certain cancer-specific proteins or genetic materials within the RBC EVs can indicate the existence of a tumor. Studies have found out that cancer cells can manipulate their microenvironment and their circulatory system, which leads to changes in the amount and the type of extracellular vesicles that's released, including those from red blood cells. In the context of diabetes, changes in the cargo of the RBC EVs, such as specific enzymes of glycation end products, can perhaps reflect metabolic disturbances that are typical of the disease. For example, increased levels of glycosylated protein within the RBC EVs can be indicative of prolonged high blood sugar levels, which is a hallmark of poorly managed diabetes. For cardiovascular diseases, RBC EVs may carry specific markers that are indicative of cardiovascular stress or endothelial damage. Elevated levels of RBC EVs containing proteins related to inflammation and thrombosis can signal an increased risk of conditions such as arteriosclerosis or acute coronary syndrome. In neurodegenerative diseases like Parkinson's, RBC EVs may be used to detect biomarkers that are linked to neural degeneration. Changes in levels of specific neural-related proteins or RNAs that are carried by the RBC EVs could potentially indicate the onset or the progression of Parkinson's disease. Alright, that is the end of this video, and in our next video, we'll look specifically at the use of RBC EVs as drug delivery vesicles.